Let's begin with our today's lecture, which is the cancer of the lower gastrointestinal tract. In this section, we will talk about the colorectal cancer, cancers of the colon and the rectum, and what's the molecular pathogenesis of the colorectal cancer, how it develops and how it progresses. So lower gastrointestinal cancers. The lower gastrointestinal cancers include malignant tumors of the colon, then malignant tumors of the rectum, and also malignant tumors of the anus. So lower gastrointestinal tract is mainly three structures. We have the colon, then we have the rectum, and then the anus. So colon is divided into four parts: ascending colon, transverse colon, descending colon, and the sigmoid colon. After the sigmoid colon, there is a rectum, which is the storage place for the fecal matter, and then we have the anus through which the fecal matter passes out. And we have anal sphincters also. We have internal anal sphincter, and then we have external anal sphincters. As with any sphincter, these sphincters play an important role in the opening of the cavities, and when they are relaxed, they open, and the fecal matter passes out. So all the cavities are basically controlled by these sphincters, and when they open, they let the passage of the fecal matter, and when they close, so different pressures, different diseases can affect these sphincters and cause fecal incontinence. So we have tumors of the colon, then the rectum, and the malignant tumors of the anus. All these structures, if you see, these are the different growths of the tumor. This is the rectal tumor. These are all the pictures of the different structures, including we have the tumors of the colon, then the rectum, and this is the anal tumor. Colorectal cancer, these are the polyps. Colon polyps are the common pathology, and when the polyps are present, these polyps have the tendency to become malignant or undergo malignant transformation and develop into the cancer. So polyps are the mucosal growths, and polyps can be sessile polyp, or they can be pedunculated polyps. Sessile polyps do not have any stalk or attachment. Here, if you see, these are the pedunculated polyps. They have the stalk attached to that attaches them to the lining of the intestine, colon. So polyps, these are the colon polyps. The molecular pathogenesis of the cancer in colons, it's about 30% of the colorectal cancer is in the middle age group. So this is 30% of the colon cancer are in the middle-aged individuals. 50% of the colon cancer, they are in the elderly people. So 30% in the middle age group, and then 50% in the older population, elderly people. But it's not a disease of the young individual. Colon cancer occurs little late in the age. Less than 1% of polyps become malignant. So these polyps have the ability to become malignant. from adenomatous polyps. So the polyps that have ability to develop into malignancy are mainly the adenomatous polyps, or the now all of you know what adeno is. I keep on repeating this word. So the polyps that develop from the glandular tissue. Adeno is for the glands. So adenomatous polyps that develop from the glandular tissue, they have more tendency to develop or grow into malignancy. So these are the colon polyps, and when this is the colon tumor or the cancer. Here again, this is the growth polyp, and these polyps even can become cancerous. So again, molecular pathogenesis is what are the changes that occur at the molecular level in the genes or in the different molecules, what the mutations occur that can be lead to the carcinoma of the colon. So first is there is point mutation in KRAS proto-oncogene. So there is proto-oncogene which known as KRAS. There are mutations or changes in that KRAS gene that leads to the development of the colorectal cancer. Here if you see very nice picture of the colon, this is the ascending colon as the going up. Ascending means going up. So ascending colon, then we have the transverse colon, descending, going down. And the last part is the sigmoid colon that becomes or at the end enters or ends at the rectum. So this is the growth or the tumor you can see in the last part of the colon or close to the at the end of the descending colon or the sigmoid colon. So there is point mutation in the KRAS proto-oncogene. Hypomethylation of DNA. DNA is deoxyribonucleic acid, and there is hypomethylation, or hypo is decreased level or hypomethylation of DNA. Then we have allelic loss of APC and GCC genes. So these genes are also, there is loss of these genes that can lead to the colon cancer. And then there is altered proliferation of mucosa. There is alteration in the proliferation of the mucosa. Poorly differentiated cells are produced from the mucosal lining, and this can lead to the colorectal cancer. Then we have progression to polyps and carcinoma. With that alteration in the mucosal lining, we can have either the formation of polyps are formed, which are the outpouchings of the mucosal lining. When the mucosal lining become hypertrophy or grow or proliferate, they lead to the formation of polyps, and also cancerous growth can occur. So polyps become also cancer, but mostly is the adenomatous polyps. So these are some changes that occur at the molecular level, at the lower level, and molecular pathogenesis mainly involve changes in the different genes. We have changes in the DNA, which is mainly hypomethylation or decreased methyl levels, and then there is alteration in the gene or loss of APC and DCC genes, and also altered proliferation of mucosa. So all these are the changes at the molecular level which progress to the colorectal cancer. Polyp conversion into cancer. So we already mentioned that the polyps can also progress or develop into cancer, and these polyps conversion mainly depends on the gross appearance of the lesion, how the polyp grossly or with the naked eye it looks like or how it appears. Here if you see, this is the cecum, then we have ascending colon, transverse colon, descending colon, and then the rectum and the anus, and these are the polyps. So many polyps present in the mucosal lining of the colon. All these are the polyps. So many polyps are present here. So how the gross appearance of the polyp is, that mainly tells us whether the polyp will change into the cancer or not. Histologic feature and size is another important feature. Histology and size is important. If the histology shows different mutations, if the histology shows proliferation, and if the histology shows the presence of poorly differentiated cells, poor differentiated cells are associated with malignancies. They will have ability to change in the cancer. So those polyps who have histologic picture and the features and size, they all depends if the polyp will change into malignancy or not. Then pedunculated or sessile, this is also an important factor. Depending if it's pedunculated or sessile, that it will change into malignancy or not. Tubular villus or tubulovillus. So all these are the different types of polyps which will have the more tendency to grow into malignant cancer. So polyps can change into malignancy. Polyps in the colon should always be followed properly, should always be biopsied to look for any changes that can lead to the development into cancer or malignancy. And then we will talk about it later on in our lecture also, that polyps in the colon are common. They are present usually in the colon, but as we mentioned, less than 1% become malignant, and usually the one adenomatous polyps can have more ability or tendency to develop into the cancer. And then we have some factors associated with it depending on the size, gross appearance, whether it's sessile or pedunculated, whether it's tubular villus or tubulovillus. All these factors can lead to the progression of the polyp into the cancer. And the detectable after more than 5 years of growth, and usually by the colonoscopy. So colonoscopy by examination of the colon is usually help us find these polyps, and after 5 years, more than 5 years of growth, usually they are detected. And the method of detection is colonoscopy, when you examine the colon with the scope directly and visualize inside of the colon. So that was all about section 1. Thank you for watching sqadia.com.